My presentation is about phase synchronization in an oscillatory network and response time variability during a sustained attention task. Sustained attention is usually evaluated using continuous performance tests, which are tasks with the repetitive presentation of similar stimuli. These stimuli are visual or auditory or both of them. Some stimuli are defined as target and some of them as non-target. For several minutes, these stimuli are presented pseudo-randomly. Individuals are requested to press a button as soon as a target presentation and don't press the button while non-target are presented. Average and variability of correct response time in different trials are an important measures in CPT tasks, and these measures are usually used for diagnosis of attention disorder and is considered a significant cognitive performance index. This figure shows a typical response time recorded from a normal children. Although intra-individual response time variability has been studied for a long time, a neurocognitive reason and origins for such changes in response to the same stimuli are not fully understood. So far, this variability has been attributed to several factors, such as brain connectivity, maturation, or some dissociable latent brain state features. This study aimed to show how the phase synchronization phenomena frequently reported during different neurocognitive tasks can cause brain response variability. The CPT that we used for gathering human data and also for simulating the suggested mathematical model contained 400 trials. In each trial, number 1 or 2 presented visually or auditory. Top figure shows one blocks of 100 trials of the experiment that each of the stimuli are presented in a pseudo-random way, visually or auditory. Individuals were requested to press a button as soon as the presentation of target stimuli that are visual or auditory 1, and do not press the button while non-target stimuli are presented, which are visual or auditory 2. The instruction was given to the individuals before the test. Therefore, individuals have to keep this information of target and non-target stimuli in their short-term memory till the end of the task, which takes about 20 minutes. This slide shows the total part of the model designed and implemented to mimic human behavior in the mentioned CPT and some other extended version of CPT, which also contain distractors in addition to target or non-targets. In order to save the time, I just focus on explanation of 2 blocks of this model. Because response time variability, which is the topic of this article, is mostly originated from the function of these 2 blocks. More detail about other parts can be found in my book titled Neurocognitive Mechanism of Attention. Actually, these 2 blocks work based on the concept of adaptive resonance theory proposed by Stefan Grasberg and Gail Carpenter. These 2 blocks contain a number of oscillators, each of which can resonate with a specific frequency, similar to the population of neurons that have a specific field to a certain characteristics. In this model, we coded each stimulus with a specific frequency. Therefore, each stimulus can activate a specific oscillatory unit in the first layer. Activated unit can resonate with one of the existing units in the second layer, I mean short-term memory. Units in short-term memory keep the information of target and non-target stimuli that the individuals were expected to keep in their memory till the end of the task. Activation of each unit in second block determine the final reaction. For oscillators, we use Van der Pol equation, which is easy to implement and interpretation, and also used in neuronal modeling in previous studies. Left box shows the mathematical formula of this oscillator. In this equation, x is the state variable, y is the output activity of each unit, correspond to the neuronal activity. Lambda is the bifurcation parameter. When its value is lower than 0, there is no oscillation. For values higher than 0 and lower than 1, it oscillates by the frequency that can be determined by the value of p. And for values higher than 1, harmonics appears in the output. To avoid complicated behavior and easier interpretation, we keep the lambda value lower than 1. To couple different oscillators, the output of the other oscillators can be added to the state equation by the coupling weight shown by capital B in this equation. If we consider that the second oscillators oscillates with the frequency of omega, in order to have synchronization or resonance, coupling weight, capital B, should be in highlighted regions, and in other regions there is no synchronization. However, within the highlighted regions, variation of B value can affect the final response characteristic that we'll discuss later. This slide shows the output of one layer to a specific stimuli. In this example, visual 1 presented from 20 to 90, the sample of time, shown by dashed dotted green color. To the push of the response of different units in a layer is plotted using different colors. By push of the activity, I mean the push or envelope of the oscillatory activity of each unit. As shown in this figure, by the presentation of visual 1 unit with the frequency sensitive to visual 1 activated or resonance. This figure shows changes of responses when a stimulus is presented in a shorter period. After the onset of the stimuli and synchronization, a period of desynchronization and decline of activity can be observed. Such pattern is also observed in neuronal or EEG recording. This is called event-related desynchronization, or ERD, which usually appears just before event-related synchronization, or ERS. Based on the model, ERD happens because of non-alignment of the initial phase of oscillators in the time they try to synchronize with each other, which this non-alignment can be different trial by trial. This slide shows push of the activity of two mentioned layers into a partial set of stimuli presented during the CPT. The first row shows the onset and duration of different stimuli, each of which represented by different color. The second row represents the push of the activity of units in the first layer, and the top row shows the push of the activity of units in the second layer. We can consider the completion of the synchronous period as the beginning of planning for response. As can be seen, the synchronous period and also the desynchronous period is different in various trials for the reasons mentioned before, and this causes the completion of synchronous period to be different across trials, even in trials with the same input. As a result, we will have intra-individual variability in response to stimuli in a CPT. The value of B, the coupling weights between oscillatory units, can affect characteristics of ERD and ERS. In one of our studies, we showed that increasing the coupling weight increases the ERD and ERS amplitude in a logarithmic shape and decreases the ERD latency or duration. Our data recorded from 3 groups of normal young adults, typical developing children, and children with diagnosed attention deficit disorder, ADD, showed that variability of RT values in ADD children is higher than typical developing children, and in children is higher than normal young adults, which are shown by blue bars in this figure. The proposed model was also simulated using the same input test given to the human participant, and model response time variability was recorded considering different values of B, the coupling weight. As you can see, to mimic human data, models of adults have higher value of B in comparison with the models of children. And the models of children with ADD have lower value of B in comparison with the model of typically developing children. These results showed that higher intra-individual response variability in children with ADD compared to typically developing children may be because of different value of coupling weight between neurons, which is somehow in agreement with connectivity-based studies, but also needs more future studies and designing experiments. The other conclusion of this study is that the phase synchronization phenomena has a role in brain response variability according to our mathematical model, and coupling weight between oscillatory units, the neuronal units connectivity, can affect the brain response variability. I want to take this opportunity to thank psychologists and psychiatrists of ATIA clinics of Neuroscience and Neuroscience Center for their kindly help and procedures of experiments and providing the required permission for carrying out experiments. And special thanks to teachers of Kosar Primary School for their help in arranging the experiments. And also thank the children parents for their patience during the experiments. I would also thank Amirkabir University of Technology for providing prizes and giving us the opportunity to gather data from normal adults, and thank students of Amirkabir University of Technology for voluntary participation in our experiments. And also a special thanks for Professor Farzad Toidkha and Professor Reza Rostami for their supervising of this study and also their help for gathering data. Thank you very much for your attention.